Hello, everyone. My name is Catherine Heldy, and I'm the Chief Scientific Officer here at Syngap Research Fund. Welcome to today's webinar. It's April 14th, 2025, and our current priorities are all about clinical trial readiness, including understanding the progression of SYNGAP1 related disorder in our patient population and having validated clinical measures to chart the progress of disease. We feel urgency about this so that we'll be ready to test new medicines that are in development with various pharma partners right now. I'm grateful to be able to introduce today's presentation. The title is SYNGAP1 ProMMiS, Natural History and Clinical Trial Readiness. And so it's my absolute pleasure to introduce today's speaker, Dr. Jill McKee. Gillian McKee, MD, PhD, is a neurogeneticist and epileptologist at Children's Hospital of Philadelphia, which we usually call CHOP. She's a provider with the Epilepsy Neurogenetics Initiative, called ENGINE, and her areas of expertise are epilepsy neurogenetics and SYNGAP1 related neurodevelopmental disorder. Dr. McKee and her team at CHOP have seen the largest number of Syngapians in the US. She gave a summary of results from the first year of the natural history study at SRF SYNGAP1 Science Conference a few months ago. You can view that on our website. It's webinar number 96L. Dr. McKee has since won a prestigious early career award at the massive AES conference called the Susan S. Spencer, MD, Clinical Research Training Fellowship in Epilepsy, which was active for the years 2024 through 2026. That award is for her research project titled Deep Phenotyping and Biomarker Discovery for Clinical Trial Readiness in SYNGAP1 related disorders. In addition to her extensive training and accolades, Dr. McKee is an exemplary team member. In fact, she's everything we want in a collaborator. She's smart, open to new ideas, generous, and she's universally liked, even by the most discriminating individuals, our own beloved Syngapians. A recorded version of this webinar will be available on the SRF website under webinars, under the news and events menu. By the end of this presentation, you will have the opportunity to get the answers to your questions. We'd love to hear from you. Please write questions in the Q&A below. For those of you just joining us, welcome. And again, our speaker today is Dr. Jill McKee. Her presentation is SYNGAP1 ProMMiS, Natural History and Clinical Trial Readiness. It's now my pleasure to turn things over to Dr. Jill McKee.
All right. Thank you so much, Catherine, for that very, very kind introduction. As Catherine mentioned, I'm going to be giving you a brief overview of our first year data from our ProMMiS Natural History Study, as well as some motivation for the work and then future directions and things we're working on currently. So objectives for the talk today is I'm just going to review some of the foundational work that led up to our natural history study and helped inform some of the outcome measures we are currently exploring. Discuss our overall protocol design for our first large prospective multi-site natural history study into SYNGAP1 related disorders. And then give a broad overview of the first year of data from our natural history study, including seizure histories, results from our developmental clinical scales, some QEEG biomarker discovery information, brief look at genotype-phenotype correlations, preview of some patient or parent-reported outcome measures, and then also discussing our future directions and what our plans are for the rest of the study. So as you guys are all well aware, SYNGAP was first discovered in 2009, so it's been around for a while and is among the most common genetic developmental and epileptic encephalopathies. It's characterized by developmental delay, autism, intellectual disability, and generalized epilepsy. And it's actually one of the few monogenic causes of generalized epilepsy, including myoclonic astatic epilepsy. And despite significant research interest into this, as I'll show you, genotype-phenotype correlations remain slightly elusive, but I think we are actually finally making some significant progress there. So I'm not intending this to be a comprehensive literature review, but just want to broadly give you guys a sense of what's been out there and what's in the existing scientific literature. So this is the 2019 study by Vlaskamp and colleagues. And they described the phenotypic spectrum in 57 individuals with SYNGAP1 related disorders. And they looked in depth at the epilepsy phenotype in particular, showing that generalized seizures, especially epilepsy with eyelid myoclonia and epilepsy with myoclonic atonic seizures, were the most common. And they also looked at comorbidities, showing not only the almost universal intellectual disability, but really highlighting the behavioral issues that I know so many individuals struggle with. In addition to general descriptions of clinical features, we naturally want to know how the specific change in a specific individual affects their disease course. So this 2016 study by Mignon and colleagues tried to address this question and found that in their admittedly very small cohort of 17 individuals, that those with variants in exons 4 or 5 had seizures that were more pharmacoresponsive, so responded to seizure medications better than individuals with variants in exons 8 to 15. So this was a very promising start to looking at genotype-phenotype correlations in SYNGAP almost 9 years ago now, but it's still, we haven't actually made much progress since then. And then there's this recent paper in Epilepsia by Waltrout et al. Looking at the citizen health registry. They looked at data from 147 individuals with SYNGAP1 related disorders and looked broadly at the most common phenotypes. And they also highlighted some genotype-phenotype correlations, showing that individuals with variants in exons 1 through 4 may have more mild presentations. So as you can see from this blog post by one of our genetic counselors, Natalie Ginn, changes in the SYNGAP gene are very broad. They range from single amino acid substitutions to protein truncating variants to whole exon, multi-exon, and even multi-gene deletions. And while we know the overall mechanism of the disease is loss of function or haploinsufficiency, meaning that just one copy of the gene isn't working, so you presumably have about half the normal or typical level of SYNGAP protein, teasing apart the specific genotype-phenotype correlations has been difficult. So finally, the last prior study I'm going to review is actually one that from our group that is just coming out in Genetics and Medicine this month, where we integrated data from multiple different retrospective sources, including the Citizen Health Registry, individuals followed at our local institution, and then from insurance claims data, because luckily SYNGAP has an ICD-10 code, so we're able to pull information on a large cohort of individuals with SYNGAP from insurance claims data. And what we found is that similar to prior reports that seizures tend to be generalized, but also that they tend to occur later than in other monogenic epilepsies. So here I'm showing you on the bottom, seizures reconstructed in monthly time bins across the first 5 years of life. And redder or darker colors means more frequent seizures, and yellow or lighter colors means less frequent seizures. And what you can notice is that kids with SYNGAP tend to have very frequent seizures. There's a lot of red, but they tend to start later, typically after age 2 or 3, which is in stark contrast to some other genetic epilepsies, such as STXBP1 and SCN2A and 8A, which I'm also showing for comparison. We can also, using both the Citizen Health data set, our local electronic medical records and insurance claims data, pull medication prescriptions to see what seizure medications and what behavioral medications are most commonly prescribed for children with SYNGAP. And what you see here for seizures is that there's a lot of Keppra, Depakote, and then Onfi early on, followed by more use of lamotrigine a little bit later. And then for behavioral medications, you can see that they tend to be a lot of meds for sleep early on, transitioning more to a lot of antipsychotics and medications for behavioral indications later in life. So when you have both detailed reconstructed seizure frequencies over time and start and end dates of medication prescriptions, we can perform a comparative effectiveness analysis to try to see which medications are more likely to either reduce seizure frequency or maintain seizure freedom. And what we found with this analysis was that both valproate or Depakote and lamotrigine or Lamictal stood out from the rest of the seizure medications in terms of effectiveness. The benefit of claims data is because you have access to very, very large populations. We can also compare clinical features in SYNGAP to other broad epilepsy and neurodevelopmental disorder cohorts. So here I'm showing features in the frequency of different features in SYNGAP on the y-axis compared to a very, very large, almost 700,000 individuals with what we call like a G40 plus diagnosis. So it's a broader epilepsy cohort. And what you can see is that developmental abnormalities, behavior abnormalities, and autism and intellectual disability stand out as much more common in individuals with SYNGAP. We could also compare SYNGAP to other more similar syndromic comparisons. And here I'm showing you Angelman syndrome and Rett syndrome. And again, you can still see that behavioral concerns and autism are much more common in SYNGAP than even in these other two groups. In the prior graph, I was just showing statically collapsing across time the overall frequency of these different features, but we can also look longitudinally across the age span to see when do these features really distinguish themselves in individuals with SYNGAP. And we can see for the behavioral abnormalities and the autistic behavior that these features tend to diverge in about the second year of life in individuals with SYNGAP and really become distinctively enriched in that cohort. And then finally, the citizen data set is really great for looking at milestone acquisition. So here in panel B, I'm showing you just the overall proportion of individuals in the citizen data set that have achieved a specific milestone. And you can see that it really ranges from about 20% in the ability to use verbal communication to more than like 95 for a lot of like gross motor skills. And then in panel A, I'm showing for the milestones that were achieved, for the individuals that did achieve a specific milestone, what time course did they achieve it? And again, you can see that like ability to use short phrases is something that is achieved much later than more gross motor skills. So while this retrospective data is very rich and does give us a lot of insights into the natural history of SYNGAP, to really think about designing clinical trials, we need well thought out prospective natural history data. So we have created this GCP compliant framework for our natural history study and consortium, where we have multiple study sites, a data coordinating center, and then we're interacting with advocacy organizations, industry partners, and other researchers to share our data. So a brief overview of our actual protocol. So we started in July 2023 with a goal to enroll 100 individuals with SYNGAP, and we have already exceeded that. So we're very, very happy with that progress. And our first part of the study was just cross-sectional, wanted to see as many individuals across the age span as possible. And then the second part was the longitudinal part, where we wanted to see individuals back every 6 months to get repeat assessments to see the stability of these assessments and how what the natural trajectory of performance on these different scales is over the lifespan. And then we're also performing quantitative EEG, going to start doing visual and auditory evoked potentials, also some other sleep and movement monitoring as well. We are also asking families who participate in the study to fill out some parent-reported outcome measures, surveys, and these assess different domains such as daily functioning, communication, sensory profiling, processing, quality of life, adaptive behavior, sleep, seizures, autism, and GI features. We really want to see if we can, how much of the spectrum of SYNGAP can we measure just with parent questionnaires versus how much do we really need to get children back into the clinic every 6 months to perform these standardized developmental assessments. And then here's the list of the actual clinical assessments we're performing in clinic. So they assess gross motor, fine motor, cognitive and social development, language and communication, and features of autism as well. And this is based in an pretty much lasts a full day clinic appointment where you have evaluations by neurology, developmental pediatrics, physical therapy, occupational therapy, speech therapy, and genetic counselors. So what have we learned so far? So this slide is actually already out of date because I can't keep up because we're seeing so many individuals, which is great. And so much thanks to the advocacy from SRF and helping connecting families with us. But so this is as of, I think last month we'd seen 98, but now we're over 100 individuals. Split sex differences about 50-50, and we're seeing people really across the whole age span, as young as 1 year old up to 68 years old. And we're also seeing individuals from all over the country. This is a map showing where, it's a little outdated as well, but showing where a lot of our patients have come from. And we've also seen people from Canada and Argentina as well. And just highlighting our 3 ProMMiS sites here. So we are over here in Philly at the Children's Hospital of Philadelphia. We also have Megan Abbott at Children's Hospital of Colorado, who's been seeing patients as well. And then we're starting up a Stanford site as well with Juliet Knowles. So here's just a kind of cartoon picture of the variant spectrum for individuals who have participated in our natural history study. And what I'm showing you is kind of the genes stretched out there in the middle and the protein truncating variants. So those are like nonsense variants and frameshift variants are on top. And the missense variants, excuse me, are on the bottom. And what you can see, which is consistent with what's been previously reported in the literature, is that most variants in SYNGAP tend to be, most clearly disease-causing variants in SYNGAP tend to be protein truncating variants. Okay, now I'm going to briefly show you a little bit about the epilepsy histories. So this is similar to that figure I showed you from our retrospective data analysis, but this is instead for the first 56 individuals in our natural history study who have seizures. Showing, it looks actually very similar. You notice lots of red, which means lots of people with many seizures per day. And then you notice seizures tend to start after age 2 or so. And then once they start, the proportion of individuals who have seizures tends to stay about steady over time. And contrast this with another common genetic epilepsy, SCN1A, and you can really notice a stark difference in that seizures in SYNGAP, when they happen, tend to happen more frequently, and that's not unexpected because of the types of seizures SYNGAPians tend to have, which are like absence or myoclonic or eyelid myoclonia. Those types of seizures tend to happen many, many times per day. And then you also notice here the relatively later onset of epilepsy in SYNGAP compared to SCN1A. So let's move on to our developmental scales. So the first one I'm going to show you is the GMFM, which is a scale that was designed to assess gross motor function in children with cerebral palsy. And it's designed to test the equivalent skill sets of a typically developing 5-year-old. So that would be a score of 100. And it's not a norm reference scale. Children are compared to themselves over time. And what I'm showing you here is, and this is how I'm going to display all of the developmental scale data, is just focusing in on the age range right now up to age 10, because that's where we have the most number of patients and see the most consistent developmental trajectories. But you can see each individual's baseline visit their score is displayed in red. Second visit is in green, which should be roughly 6 months after the first one. Third visit in blue, and then fourth visit in purple. So we do actually have some people who have now come to see us for 4 visits, which is really great. And then individual visits for a specific patient are connected by a line as well. And what you can see here is, well, we don't think of gross motor function as one of the most severely affected domains in individuals with SYNGAP. We do see a very nice developmental trajectory over time. It's not like all the kids are sealing out on this assessment. They're not all scoring 100s. There is definitely significant measurable impairment, but you do see a nice developmental trajectory over time, which is important when you're considering an outcome measure for a potential trial, because we want to know what the typical natural history of SYNGAP would be, so that if we were to, for example, give a child a gene therapy, we want to be able to measure, is their new trajectory different than what we would have expected without having given them the intervention? So next, I'm going to show you the results from the Bailey-4. So this is a norm reference test designed for children from 0 to 42 months of age, but we're performing it on all comers in the study. And it assesses development in 5 different subdomains: cognition, expressive and receptive language, and fine and gross motor. So here I'm showing you the motor subscales of the Bayley. So gross motor on the left and fine motor on the right, displayed in the same format as before. And what you can see here is, as well, there's actually a pretty good developmental trajectory over time. There's not a ton of fluctuation up and down for individual patients. And we're not seeing a lot of floor or ceiling effects either. Now contrast that to the cognitive and language subscales, which is oddly what I would consider to be some of the going into this, I thought these would be some of the more important scales to look at, because I think of language and especially expressive communication as significantly impaired in individuals with SYNGAP, as well as cognition. But it's not actually the most reliable developmental trajectory. You can see kids really do span the gamut of scores on these assessments. And also even across assessments in the same child, they're not necessarily consistent. So for example, I'm highlighting this one individual here where you could see on their first assessment, they scored about an 80. Then on their second, they were like a 30. And then on their third assessment, they were back up to an 80. And we actually went back and reviewed the clinical notes for that assessment. And it seemed like this dip in their score was actually attributable to their behavior on that clinic day. And we all know that behavior can be a significant issue in children with SYNGAP. So it's not that unsurprising, whereas the gross and fine motor skills are more easy to measure consistently across sessions. And then finally here, I'm showing you the P-body, which is a norm reference test for children from 0 to 72 months of age. And we're performing the fine motor subsets only, so looking at hand manipulation and eye and hand coordination. And here as well, similar to the motor subdomains of the Bayley, you can see that there is a pretty consistent developmental trajectory over time. So moving on to quantitative EEG. So one part of our study is that each time an individual with SYNGAP comes to clinic, we're performing not a clinical EEG where you're looking, epileptologist is reading it to look for seizures or look for spikes or discharges to help support a clinical diagnosis of epilepsy. For these research EEGs, we're actually performing computational analysis, looking at like the spectral features of the EEG to see if we can identify a biomarker for SYNGAP. So just a couple quick differences about these types of EEGs. Our high density research EEG has a much higher sampling rate. So we're sampling that signal many, many more times per second than in a clinical EEG. It also has many, many more electrodes. Clinical EEGs have 19 scalp electrodes. Our high density one has 128 scalp electrodes. And it's a really nice net or cap-based system that you just have to put on the individual's head and kind of like wet their hair a little bit so the electrodes make good contact. So it's much easier to apply than a traditional clinical EEG where you have to glue each electrode onto a child's scalp. And we found it's been very well tolerated for our study. And kind of proof is in the pudding. We've been able to collect now over 120 quantitative EEGs on our individuals with SYNGAP through our natural history study, which is great. And that's including 71 unique individuals. Just here comparing the cohort of SYNGAP EEGs to another neurodevelopmental condition, STX-BP1, and then also to all of our control EEGs, and they span the lifespan. So what can we do with this EEG data? So this is work that was done by Peter Gaylor, who was a postdoc with our group. And what he did is he trained a machine learning model to predict a diagnosis of SYNGAP using spectral EEG features. So different, it's like the power of different frequency bands to try to predict whether that EEG was from someone with SYNGAP or from a healthy control. And I'm showing the ROC curve here, showing that he was actually pretty good at being able to predict a diagnosis of SYNGAP. But what's really cool about this data-driven analysis technique is then you can pull out which features the model used to predict SYNGAP. So which features of the EEG were most strongly able to predict that that child had SYNGAP. And that's what I'm showing you over here. And the most strongly predictive feature was actually the alpha theta frequency range in the occipital region, so in the back of the head. So then what we can do is pull out that specific EEG feature. So this is showing the occipital alpha theta power in 3 different age bands, because one important thing about EEG is it does change across development. So that's why we've been individuals by age here. And you can see in all 3 categories, toddlers, children, and adults, there's lower occipital alpha theta power in individuals with SYNGAP compared to neurotypical controls, which is really exciting for a potential biomarker of SYNGAP. And the other really exciting thing is Peter initially did this with clinical EEGs. So those that were obtained for clinical purposes for children who were followed at CHOP who happen to have SYNGAP. And that's what we're showing you here in the green bars. But then we were able to confirm this in completely different cohort in our research quantitative EEGs. So that's what I'm showing you in blue. So these are different individuals. It's a completely different recording system, as I showed you previously. And we're still able to find that this result holds, which is pretty exciting. All right, so the next thing I'm moving on to is back to genotype-phenotype correlation, which I kind of alluded to a little bit at the beginning of this talk when I was reviewing what's been previously published out there on SYNGAP. So if you remember, the Mignon study from 2016 showed that seizures in individuals with variants in exon 4 to 5 seem to be more pharmacosensitive, so more responsive to seizure medications than other variants in later exons. This paper here showed that there were milder symptoms that were observed more frequently in individuals who had variants in exons 1 through 4. As did this Agarwal paper, again, showed milder phenotypes were more frequently seen in individuals with variants in exons 1 through 4. And then the Wildtrout paper as well, most recently from 2014, showed that those with exons 1 through 4 variants were more likely to have the ability to speak in phrases, and also found that epilepsy occurred less frequently in patients with variants in the SH3 binding motif. So some exciting work showing hinting at the potential of genotype-phenotype correlation, but people have been talking about this for almost a decade, and we still don't have a very clear idea of how does this actually affect an individual patient, and why does this occur? So the simple why does this occur is, we think, isoforms. So when you make a protein, there's many different ways to do it. It's not, we think of like, you have your coding sequence and that's translated into protein and that's your protein, but it's actually much, much more complicated than that. You can build a SYNGAP protein by taking various combinations of these building blocks, so these different exons, and putting them together in slightly different ways to make all of these different proteins. So theoretically, if you had a variant in one of these exons that wasn't used in a version of SYNGAP that was highly expressed in the brain, it's possible that you might have a milder phenotype. And that's what we think is going on, especially with variants in exons 1 through 4. Because we found that there are isoforms of SYNGAP that start after that point. So if you have a variant early on, even a protein truncating variant that cuts off the protein, if you're able to make SYNGAP starting after that point, you could still make that isoform and have it be expressed in the brain. So by that logic, people with early variants may actually not be completely haploinsufficient. They may have some intermediate level of SYNGAP. So we know there's all these different ways you can build SYNGAP, but what's actually expressed in the brain? And that's something we're still working on trying to figure out and is a little bit tricky. But from GTEx, which is a publicly available expression database, we can see that there's at least several different isoforms of SYNGAP expressed in the brain. All of these little yellow bars are different parts of the brain. And you can see by the darkness of the circle how much this specific isoform of SYNGAP is expressed. So it's definitely not just one. And then what we've done too is we have a cohort of individuals who've undergone epilepsy surgery at CHOP. And when they undergo epilepsy surgery, a small sample of brain is resected as well and sent for sequencing. And that's what I'm showing you here is the expression level over here in transcripts per million of these different isoforms of SYNGAP. And what we can see is that a lot of them are expressed, and especially this one that seems to be the most highly expressed in this sample of individuals actually doesn't include the first 3 exons, which is very interesting and makes me even more curious about the exon 1 through 4 variants being milder, because if you can still express this variant, this version of SYNGAP, if you have a variant in exon 2, for example, that would be really exciting. Here's just another database of SYNGAP expression, looking at fetal brain expression. So not necessarily corresponding to what you would see in a child or an adult, but again, many, many different isoforms of SYNGAP that are expressed at least to some level in the brain. So going back to our variant spectrum in our natural history study cohort, we wanted to see how many individuals we actually have with variants in the first 4 exons. And it turns out, as of this data freeze about a month ago, we have 12 individuals with first 4 exon variants. 4 of them were splice sites, 4 of them were frame shift. 3 were a small deletion that occurred from the start of SYNGAP through exon 3. And then 1 was an intronic deletion insertion. And these individuals also span the age range from as young as 4 years old up to 47 years old. And then just one other piece of evidence and exciting preview is this is from a paper done by Alicia Harrison, one of our GCs who specializes in SYNGAP, looking at inherited cases of SYNGAP. And this was actually just accepted for publication this morning, so this is a preview. It should be out soon. But we looked at 5 different families where the SYNGAP variants were actually inherited. And of the 5 families, 4 of them were actually in the first 4 exons, which maybe lends some other credibility to the hypothesis that these may be slightly milder SYNGAP cases, at least mild enough for these individuals to grow up and reproduce. So let's get into the data now. So first, I'm going to show you some of the classification scale data. So this is stuff I didn't show you for the whole broader cohort, but for both the gross motor function classification scale, which is the GMFCS, the mini and the max, so mini, max, and max, which is the manual ability classifications system, and then the communication function classification system, which is a scale of communication. I'm showing you the mean and median scores for those with early variants, which is those with variants in exons 1 through 4, and late variants. And on this scale, lower scores mean improved functioning. And what you can see is that for all 3 scales, there are significant differences in the scores for individuals with early variants compared to everyone else, which is exciting. So then if we get into the actual more detailed clinician-assessed developmental scales, here I'm showing you the GMFM. So that's a scale looking at gross motor function. And here, instead of color coding individuals by the visit, so by the encounter, everyone in green here has a SYNGAP variant after exon 5. And then the individuals I'm showing you in red have exon 1 through 4 variants. And then there's also a couple of blue individuals, which we won't get into now, who have something other. So I will tell you, one of these is a SYNGAP duplication, actually, and then one is a missense variant that we actually do not believe to be explanatory for this individual. So that person likely does not actually have SYNGAP. But what you can see is that the red variants, so for those with variants in exons 1 through 4, these individuals all tend to be at the milder end of the spectrum. So if you recall, a score of 100 is a score for a typically developing 5-year-old. Then here I'm showing you the Bailey scores as well, and you see the same pattern kind of across the board in all 5 subsets. You can see that the red individuals, or the exon 1 through 4 individuals, all tend to have higher performance on all of these different subscales. And then looking at the CARS, which I also didn't show you across the whole population, but this is an autism type scale where it assesses 15 areas of behavior to help identify individuals with autism. It's not actually a diagnostic test for autism, but it tells you based on their score how many symptoms that could be consistent with autism they have. So higher scores here are more symptoms of autism or severe symptoms of autism. If you're over 35, it's considered consistent with severe symptoms of autism, or sorry, not over 35, over 30 is symptoms of autism. And what you can see here is almost all of the individuals with variants after exon 4 have symptoms consistent with autism in green, whereas our exon 1 through 4 individuals tend to be a little bit milder, or at least more of them are under that score of 30 and would not be consistent with a diagnosis of autism. And then very briefly, going back to the quantitative EEG, this is still really early and we need to look at this in more detail. But this is, again, that occipital alpha theta ratio. Grays are controls. Blue are those with SYNGAP, where the variant is not an exon 1 through 4. And the purple is individuals with variants in the first 4 exons. And while it's a little tricky because we don't have as many individuals all at within the same age span, it does seem like there is a trend that the individuals with the early variants may have higher or more typical occipital alpha theta ratios. So one other aspect of the study we're collecting are the parent-reported outcomes, which I mentioned when I went through the protocol. And while we don't have this data yet for our natural history study cohort, they're still working out the details of the contracting with RareX. We were able to pull some of this data from the Simons data set. For some of the questionnaires, we're going to be asking our families to complete as well. So that's what I'm just going to give you a brief preview of here. So this is the childhood sleep habit questionnaire, which is a parent-reported outcome measure that assesses sleep. And the individuals in green are those with SYNGAP, and then the gray dot cloud is just all other individuals with other neurodevelopmental disorders that happen to be tracked in the Simon Searchlight database. So just note, unlike the other slides where I was showing gray dots as neurotypical controls, these are not neurotypical individuals. They have other neurodevelopmental disorders. But what you can see is that those with SYNGAP tend to have pretty average scores across the lifespan, not considerably separated out from these other neurodevelopmental disorders. But you notice there aren't any ceiling or floor effects. So this could potentially be a measure that would show a change in response to a therapy. And same goes for this is the childhood behavior checklist, where on this score, if you have scores 60 or above, indicates that you are either at risk of or have clinical symptoms of behavior or abnormalities. And what you notice, most individuals with SYNGAP, especially on the later age questionnaire, the one that starts at age 16, tend to have significant clinical symptoms of behavioral abnormalities, which isn't super surprising. And then here's the Vineland-2, where on this scale, a score of 100 indicates average adaptive functioning. And what you can see here is that our Syngapians tend to cluster around like 60 initially, and then there's not much data for the older individuals, but the scores tend to be lower as well. And we have, this is the Vineland-2, we have the Vineland-3 in our protocol, but the scores are pretty comparable. And then finally, the QI disability scale assesses quality of life in 6 different domains. So social interaction, physical health, independence, emotions, leisure and outdoors, and then negative emotions as well. And here again, you can see our Syngapians tend to be fairly typical compared to other individuals with neurodevelopmental disorders, but no consistent floor or ceiling effects on this scale either. And then briefly talking about some future directions where we're going next. So in addition to EEG biomarkers, which I discussed a little bit, we're also hoping to see if we can detect the levels, SYNGAP protein levels in the CSF, and do those correlate with functional outcomes or response to therapy? Because that would be a really nice biomarker as well. Furthermore, we know sleep is a huge problem in individuals with SYNGAP. So we're starting a collaboration with Maya Buchan at Penn, where we're planning to send our natural history study participants home with one of these gene active actimetry watches to track their activity levels during the day and then track their sleep as well at night. To see how well individuals are actually sleeping through the night, which I suspect is not many of them. But this will be really helpful data as well, because potentially a therapy could improve sleep cycling as well, and that could be a measurable outcome measure. And then we are also currently making some changes to the protocol. So we've separated SYNGAP out now from our larger ENDD natural history study. It says SYNGAP is its own unique condition and has its own unique features that we need to focus on more. We're also expanding, going multi-site. So Megan Abbott has already been seeing individuals with SYNGAP in Colorado, and then we're also going to be expanding to Stanford as well. We are currently undergoing changes to become completely GCP compliant as well, and doing this in parallel with our multi-site expansion, which is a requirement for clinical trial data, so that we can also use our natural history study data as a control arm for future clinical trials. So in summary, today reviewed some foundational work that helped lead up to our SYNGAP natural history study and inform some of the outcome measures that we chose, discussed our protocol design, and then also reviewed the first year of our data, including seizure histories, clinical scales, EEG biomarkers, genotype, phenotype correlations, which is newest exciting development, brief sneak preview of our patient or parent reported outcome measures, and then touched on some future directions. So obviously this isn't just me, this is definitely a team sport. So want to thank all of our team members. We have a very large team that's making this happen. And then also all of our funding, of course. And then most important of all, need to thank all of the families who have so graciously participated in our study and keep coming back to see us every 6 months. We love you guys. You are the best. But thank you. And I'm happy to take any questions.
That's amazing. Dr. McKee, you're amazing. So I just want to say, just to remind the world, because this is one of my talking points lately, the first human was diagnosed with SYNGAP in 2009. 2009. That was like, I hope we were all alive back then, right? That was 16 years ago. And to have someone of Dr. McKee's caliber go over this much data in public is bonkers. Like, this doesn't happen. I really think we're setting a land speed record. And I think there's so many things you just said that I'm going to use those slides in conversations with parents, especially the ones who are holding 2-year-olds. They're like, they seem okay. It'll be fine. And I'm like, look at Jillian's slide. The line's going down. Get ready. Not because I savor saying that, but it's because I think people need to know what's coming, right? And now because of your work, they don't have to trust the crazy redhead. They can listen to the refined Canadian doctor who's rational and has data. This is amazing, and I can't thank you enough. How many papers did you refer to in this talk that are coming, that are on their way?
From our group, just 2. So the genetics and medicine one, which should be the final proof version out very soon.
Yeah.
And then Alicia, who I think is also on the call, our wonderful GC, has this familial SYNGAP paper that was just accepted this morning. So that one will be out soon too.
So as you know, I'm working on a blog that is way simpler than what you're talking about here. But when you think about these first 4 exons, and I've clearly gotten you thinking about them, which I'm thrilled about, I love all these slides. 2 questions. How many are out there? I mean, these are easy to miss, right? We have a small cohort in our population, not because there's a few of them, but because most of them, I think, are probably getting by without getting genetic testing. That's question 1. Question 2 is, can you talk about, every parent's dreaming about a day we have an ASO. What do you think an ASO means for these first 4 exon patients?
Yeah.
Do they get a lower dose? Do we titrate slower? Like what?
Great questions. So in terms of like the prevalence or frequency of first 4 exons, I think you're right, because as with all neurodevelopmental disorders, we are expanding the phenotype, especially on the mild spectrum, as we're doing more and more genetic testing. I'm hoping in this day and age, and it's still probably not true, but many of these individuals are being offered genetic testing. Our natural history study cohort is taking all comers. So we're not discriminating by clinical features or anything, but they do have to have a diagnosis of SYNGAP. And we're seeing a little more than 10% have, like we had 12 out of the 98 had first 4 exon variants. So it's definitely not rare, but especially among older individuals, you're right that they may never have been offered genetic testing. So again, another plug to offer genetic testing, because people always think it won't change anything by the time a person's 20 years old, but it's really helpful for us just to have a better sense of what happens over time for those individuals. And also, I mean, you guys could speak more to the parent perspective of just having an answer and knowing what the cause is, how helpful that is. And then to your second question about what does this mean for clinical trials, we don't really know. It's promising that if these first 4 exon kids are not quite haploinsufficient, like they're somewhere in between, and we're able to detect milder symptoms, I think that gives a lot of hope for our typical Syngapians that an ASO, even if it doesn't fully correct the levels of Syngap to a neurotypical level, that we will hopefully see some benefit, because we do see that these first 4 exon kids tend to be milder. And I really liked that you said not easier, though, in one blog post or somewhere, because it doesn't necessarily mean they're easier. We notice that a lot of times as kids develop more measurable skills, some of the behaviors can actually become more difficult to manage. So that is something important caveat as well. But in terms of dosing for them, we're not really sure. There's not much evidence, and something else I really would love to look into more is what happens when you have too much Syngap. And there is one individual that I know of with Syngap duplication who does have a neurodevelopmental phenotype, but it is very different than our typical Syngap. So I think that's an area that needs to be studied more.
That would be an amazing case study if you want to just not sleep one night.
Yeah. And if I would love to have more than one case, ideally, like a small case series would be great. So if there are any other researchers on the call who know of individuals with Syngap duplications who would like to participate in some research, would love to meet them. Because yeah, it's a sign that we may need to be careful about overshooting Syngap levels.
So 2 things, then I'll give it to Catherine. First thing, I am aware of an exon 1 patient. Actually, there's 3 things. I am aware of an exon 1 patient who has a driver's license in Europe. So that's kind of a big deal. 2, as companies work on ASOs and do talks, we are going to, hopefully they'll share this data. We are going to start seeing data where they've upregulated Syngap well past 100% in monkeys, right? So that might be interesting data to give people, because I think when I started, people were like, well, if you go over 100%, people could die. And I'm pretty sure that's not true, right? So that's exciting. And then the third question, like for McKee 2026, 2027, when you write that paper, how many subtypes of Syngap are you going to describe? Because I'm thinking there's like first 4, down the middle PTV, and then intronics and missense, which would all be sort of essentially N of 1s pending further study.
Right. It's tricky because I even think even the exon 1 through 4, we are seeing this like milder cluster, but there's not a huge gap between the clusters. They're not like clearly different, no overlap. They're definitely overlapping distributions on a lot of these scales. So it's tricky, like where do you draw the line? And then we have some later exon kids who are mild too, right? SYNGAP isn't existing in a vacuum. We're all complicated humans made of 20,000 other genes and different environmental factors that determine who we are as adults. So it's not just SYNGAP. So that's a tricky question. I feel like I haven't really answered your actual question.
No, it's a totally fair answer. That's why I gave you 2 years to write the paper. I think it'll be great. I can't wait. Catherine, I'll let you.
Yeah, I just want to tack on a follow-up from what he was just saying, which is it would be really interesting to see what are actual levels, right? What are the actual SYNGAP transcript and protein levels in the people relative to the mutations? And I don't know how likely we are to figure that out, but
yeah, it's tricky because blood doesn't necessarily mean brain, and getting CSF is great if we can get that CSF SYNGAP biomarker to work and like show differences between controls and SYNGAPians. And the first 4 exon kids would be somewhere in the middle, but it's still also not brain tissue, and it's not the SYNGAP at the synapse, and that we're not going to cut chunks of people's brains. So.
Right. And then doing the iPSC neurons, that's not necessarily the age that the brain is. So, right. So you can look at all these things, but the question is, what's the most important thing to look at? What's actually going to reflect the patient? So it's a good problem to really think about. So there are a couple questions in the Q&A, but I just wanted to say that I really, really appreciate your talk. So thoughtful, so many learnings. And one thing that really jumped out at me was on cognition and language, when you were talking about the cognition and language measures and how they jump around a bit, and how that was less consistent for maybe people's abilities. And I just wanted to point out that we see a lot of that at home too, like on a certain day at a certain hour, a kid may be able to communicate some wants or needs, and then later in the same day, not be able to do the same thing. And I was wondering if you have a sense of that being potentially impacted by absence seizures, or a kind of stubbornness, the sort of like, is this a personality thing? Do you think this is an electrophysiology thing? Or do you think that, because like if they're going to climb up the stairs, they'll often like just climb up the stairs every time. So I'm just wondering how gross motor versus communication, especially, how you think of those as complex neurological processes.
Yeah, so for some kids, it definitely could be a seizure-related thing. Like if you're having a bunch of absence seizures, you're losing time and you're not able to respond to a specific instruction because you didn't hear it because you were seizing. But I think that's the minority, because when we're doing these assessments, we're watching the kids and we're not seeing that they're having hundreds of seizures. And if they were having a lot during the assessment, that's something we'd make note of. I think it seems to us to be more autism related and behavior related. So if they're having a harder time with transitions, or they've just traveled overnight and had a rough time on the plane and they're coming to see us, that can explain some of the variability in their ability to participate. Whereas the walkings, like the gross motor, like we're just watching them walk, we're watching them do some like basic motor tasks, that tends to be less affected than their ability to participate in like a language assessment, for example, if that makes sense.
Yeah. And then also, I just wanted to follow up on Mike's question about how many different kind of disorder, subtypes are we going to end up with? And you have all this gorgeous data, kind of by test. And I'm wondering if there's some metric where you can collapse the data for a patient and then do the patients kind of head to head. So on that, and I'm not sure how you would sort of like collapse it by over test to a person, like maybe some kind of, I don't know, star chart or something of like what this looks like. But what I'm thinking is that the PTVs might all look more similar, but then the missense. So I'm thinking if you did every missense against each missense individually against all the PTVs, you would see which of them are like the PTVs and which of them are not, right? And so that initially could potentially be some kind of an indication that maybe the missense was more of a kind of closer to a haploinsufficiency and not necessarily a different disease model. I'm wondering if does that make sense to you? Is that solid reasoning, or would you want to see something else?
Yeah, I think from like in terms of clinical data, that would be a good way to get at it. But I think in terms of evaluating missense, I think our basic science or translational science colleagues may be better able to actually like expressing those variants in like iPSC cells and seeing what they look like might be more helpful. And then also some like we could do long range sequencing too to see like how much RNA is actually being transcribed from some of these kids with variants that we're not sure about how important the significance of those variants is. I haven't looked at like all missense versus all PTVs in a while. So it's something I should just take a look at again, and so we don't forget, because when I was first looking at genotype phenotype correlations in SYNGAP, that's like the obvious thing to go for that a lot of people have looked at in other conditions, because some disorders have, we call like dominant negative missense variants that have like a very different clinical picture. So I very initially looked at that in the Citizen Health data set, but didn't see any significant findings. But it's always something we should take a look back again, just to make sure nothing has changed. But our hypothesis is that missense variants and PTVs and SYNGAP work in the same way. They both lead to haploinsufficiency. They like causative missense variants. VUSs are a whole other can of worms, because some of them are real and probably do affect SYNGAP protein and cause haploinsufficiency. And then some of them may not be and may just be population variants in a child who has a different neurodevelopmental disorder, but not SYNGAP. So it is kind of tricky to tease it apart, but it's something we should keep looking at.
Right. Well, I can't wait to hear more and see the paper coming out from Alicia.
Mm-hmm.
So
I see that Suzanne asked about the older than 10 too in the chat.
Yeah. I was just opening that up. So Suzanne asked, will you eventually present Bailey-4 statistics that include more than ages 0 to 10? I think they're tested on this despite being older. So answer that, and then I'll ask a little more too.
Yeah, so we definitely do the Bailey on everyone, even though it's not actually validated in older kids. We've been doing it on everyone, and it does seem to be like kids are falling within the range. There's no floor or ceiling effects. The reason I didn't show it is the developmental trajectories get a little bit all over the place, just because we don't have as many individuals after age 10. Our highest proportion of patients enrolled are like the 5 to 10 age range. But yes, I have the data. I can definitely share it. The trajectory just doesn't look as nice.
And it looks like Suzanne Jones is being added as a panelist. Suzanne, do you want to ask follow-ups and then your second question also?
No, that's helpful. That makes sense that the data gets more skewed. Catherine, you can ask my second question for me since you've got it in front of you.
Oh, okay. So she's asking, where exactly is SYNGAP protein important in addition to the brain, like other places? So she's saying potentially endocrine function, other critical areas. What are there other critical areas that have been determined? And then, so there's that, where besides the brain, and then have the specific isoforms of SYNGAP been determined, or is this less important at this time? So
the isoform information is very intriguing.
It's really interesting. We're honestly still very early in trying to figure it out, and it's quite complicated in SYNGAP. I'm not sure if you can see this, but this is from GTEx. I briefly showed it. So these are all different isoforms of SYNGAP, and yellow is brain. So there's many isoforms that are expressed to some extent in the brain, but then all these other areas are other areas that SYNGAP is also expressed, and we're not I'm not sure at least what it's doing in all of those different areas. Some of them may be like nerve conduction related too. But it's an open question that we need to assess.
Interesting. Yeah. I know Catherine and I have talked about this in the past. I think there's an endocrine study to be done out there. Thank you very much, Jill. This is all great.
Let's raise more money and do it.
Yeah, and then Alicia's here, so let's have her talk. Oh, we can't hear you yet, even though you're unmuted.
Huh. I can see you, but can't hear you, Alicia.
Did we cover all the anonymous questions while Alicia's?
No, I'll go for those. So anonymous attendee asks, could ASO be used with missense variants?
Yes, great question. And we since we think that all missense variants do cause haploinsufficiency, they should be able to be used, because we want to increase the level of SYNGAP protein, right? And if we think a missense variant is also causing SYNGAP protein that doesn't work and is then degraded or something, then yes, the same strategies should work for most missense variants.
So I just want to offer that that's really good news, and also not necessarily the inclusion criteria for the first trial.
So that's a different question. Same as like exon 4 kids, it should work. Strategies to increase SYNGAP should work for them as well. But in an initial clinical trial, it's possible that to have the highest likelihood of trial success, people may want to include the most homogeneous, severely affected cohort, so may exclude exon 1 through 4 children from an initial trial.
Yeah, and then Alicia, did you get your
I'm not sure. Can you guys hear me now?
Yes.
Oh, perfect. Okay. Well, I don't have tons of extra detail to add on top of what Jill already covered, because she already presented everything so nicely. But yes, yeah, we just got our feedback back on the familial SYNGAP paper this morning. So hopefully that'll be out soon. And I think, yeah, we definitely don't think that familial cases of SYNGAP are like the norm. They're very much the outliers, but I think still are an important edge case to delineate as we kind of figure out where the edges of the SYNGAP phenotype are.
And would you expect that to be just because of, sort of cognition, behavior, social, these kinds of, it's just sort of a milder phenotype to, like you were saying, to be more, whether you're more likely to have a child? Is that what you're thinking?
Yeah, I mean, I think, and this is not unique to SYNGAP, in general, people at the milder end of the phenotype should be slightly more likely to be able to be in a position of having a child, just because then we start to get into, issues of consent and what people are able to say yes to. So yeah, like I said, I think this is not the norm, but it's kind of an interesting edge case, and important for us to know kind of where these edge cases lie, partly because of exactly the VUS classification, challenge that Jill brought up, which is that we have to know kind of at what point do we draw a line and say, oh, this can't be a real SYNGAP variant because it's inherited from a parent. Under what circumstances would we say, that's maybe not totally a black and white line?
I hadn't thought about that. That's frustrating. There's yet another complexity. Because we just had an exon 1 through 4 get married in Australia, by the way. Like it's happening in real time, and you're right. It's interesting. And one of the reports from parent there was things have gotten so much easier as they've gotten older, which I thought was interesting.
Yeah, I mean, and I think also one takeaway from the paper that we wrote is that we're not saying that SYNGAP is a reduced penetrance disorder. So in the scenarios where we saw inherited variants from parents, these were still parents that had symptoms. These were still parents that were affected. So I don't think we're suggesting that the end of the SYNGAP spectrum is no symptoms. But I do think that these represent an important case to pay attention to.
Yeah.
Yeah, and as alluded to, it does make variant interpretation a little trickier too, because one of like the ACMG criteria for like supporting a variant's pathogenicity is if it was de novo, meaning new in the child, not inherited from a parent. And now we have these interesting cases where they were inherited from a parent, but are still clearly SYNGAP.
Yeah, absolutely. Okay, so I'm going to move on to some more from anonymous attendee. Polygenic autism has a gender gap, more males than females. Although perhaps this is changing as diagnoses are increasing. If a gender gap still exists in polygenic autism, do you see a greater number of males than females imbalance in SYNGAP1 deficient patients who have autistic features? If not, why do you think this is so?
That's a great question. We don't see a difference gender-wise in diagnoses of SYNGAP. Our population is pretty much exactly split 50-50 males and females. We actually have not yet looked, and this is on me, at the like CARS breakdown for males versus females. And it's something I could very easily do and will do once we're done with this webinar, just to see are the symptoms different based on gender, which we have not looked at yet.
Okay, great. Thank you. Also, Paul, oh no, I already wrote that. Do you think there might, sorry, Mike?
Go ahead. Sorry.
Do you think there might be overlap in CSF biomarkers between SYNGAP1 deficient patients and Phelan-McDermid patients who Langshank 3? So that's another synapse relate, it's also in the PSD.
Yeah, that's an interesting question. So for our CSF biomarkers, we were looking at actually measuring SYNGAP levels. So that would be distinct and like SYNGAP specific. Are there other downstream ones that like downstream biomarkers in different signaling pathways that could be shared between SYNGAP and other synaptic disorders? That's definitely a possibility. It's not something we've really explored yet.
So, we have some proteomic biomarker in plasma studies going on. And so as we find stuff there, we'll definitely share that with you. And maybe there'll be some other, instead of doing like obviously it'd be great to do a full spectrum CSF biomarker study, but if that's really precious and we don't have very much SYNGAP CSF, or even any, maybe we could look at some likely candidates that might be good there.
But this is a good moment to interrupt and remind every SYNGAP parent who's watching, if your kid is going in for any operation of any type, let us know and give us time to get consents done. So if any body part or body fluid is being taken out or in, we can get some. I mean, we just had a kid who did a gut biopsy at UCSF. And if we had figured it out like a week sooner, we could have gotten SYNGAP gut to a lab at UCSF working on the enteric nervous system. So this is part of the challenge of being a record-setting gene in terms of how quickly we're moving. We keep asking families for everything.
And Mike, just to point out what Suzanne was asking about, like, we don't know everywhere. So even if you think this is not necessarily a place that has to do with SYNGAP, tell us, like, it might actually be useful and helpful.
Yeah, we have a biobank. We can store this stuff. There's two more questions that are good. You want to get to them?
Julie Miles, hi, Dr. McKee. Excellent webinar. Has N considered bringing in psychiatry to be part of the day? I know it's already a very full day, but with behaviors being such a player, as our children age, this seems to be a significant need.
Yes, absolutely. And we've recognized this from the beginning, and it's been just a CHOP logistics problem to get like psychiatry here on the same day to see kids with us has been problematic. We have developmental peds, which is great for like the younger kids' behaviors. But as people get older, we really do need psychiatry. And unfortunately, we just haven't been able to get them at CHOP. I think Colorado actually has psychiatry.
And Stanford.
Stanford, yeah.
So fortunately, this is like a CHOP specific problem.
Thank you. And then the last is from Paul Duchenko. Very nice talk. It's striking that seizure incidence comes online, so to speak, around 2 years of age. Presumably the SYNGAP protein deficit is there from birth. So he's thinking about why would it develop later?
Yeah, that's a great question. And I can't say for sure. SYNGAP, the protein is definitely deficient from birth and in utero. A lot of these proteins are developmentally regulated in that they become more important at certain points in your development. So that's possibly one reason. Another reason is because of the type of seizures that Syngapians tend to have, they tend to have generalized seizures, which require your brain to be more myelinated, to have your whole brain seize at the same time. And myelination occurs over the first few years of life too. So that could also be another reason too, not just because of the SYNGAP levels, but also just because of the rest of your brain structure. If those wires aren't insulated yet, it's hard to have a seizure coming from everywhere because they just can't get everywhere yet.
So it's interesting to think about what is happening in those 0 to 2 brains that doesn't look like a seizure because they don't have myelination. And by the way, that was an amazing answer. And it's a good time to advertise that Dr. Juliette Knowles at Stanford, who is the PI on our ProMMiS site at Stanford, also has a lab and does research focused on myelination and generalized seizures leading to worse myelination, leading to worse seizures, which we see in Syngapians. That was actually one thing. I don't know if you had a chart that showed this, because one of my talking points is always, we start with absence seizures, and then at some point kids progress to drops.
Mm-hmm.
It would be awesome to have like a CHOP level graphic that showed if it was true, that was true,
No, that's a good thing to show. Since we had early numbers only, we've been focusing just on the overall seizure trajectory, but we have the data broken down by seizure type. So those like monthly progressions we can show for absence seizures, myoclonic seizures, atonic seizures all separately, and then get a sense of that progression.
Because the follow-on question there is, assuming we had enough data, which we might not, to double click on that and say, did kids who had medicated absence seizures progress to drops later or have better developmental outcomes than kids who had untreated absence seizures?
Yeah.
Because I've got some interesting anecdotes there in my head, but the data is always better, right? And which is another, since parents are listening, you got to go to these natural history study sites, guys. Like, this data is how we figure out this crazy disease. End of lecture.
Well, you guys have done an amazing job getting over 100 kids to us so far. So thank you so much for your advocacy and getting us children and adults to come see us. And adults may actually be important too, given FDA regulations and where clinical trials may start too. So we have a lot of individuals in the 5 to 10 age range, but some over 12, more pushing the over 12 recruitment might actually be helpful for some of our pharma partners.
You can pass on this question, but there are a lot of families who have not traveled. Our families get this siege mentality and they just lock the doors and keep their kids safe, and decades go by. And then we call them and say, please go to CHOP or Stanford or whatever. And they're like, no, I can't travel with this one. How do we help those families? Like, can we give them some, can we do a telehealth and send them some like powerful drugs to knock kids out for the trip? Or like, how do we solve that problem? I'm kind of kidding, but I'm kind of not.
No, it's a really important problem. And we recognize when I've seen parents show up in clinic covered in bite marks from the plane ride, like this is a huge, very, very real problem. And a couple of things, like working with like ABA to try to work on like distraction techniques for behaviors, obviously is something everyone's doing. Medications can help. Some people have given double doses of meds before going on a plane. Not advocating, just putting it out there. And then one thing that I think is really important that would increase enrollment for a lot of these is trying to validate some remote assessments. So we've been tossing around ideas and haven't started yet of validating some clinical scales that you can do via telehealth, like the DAISY, which is similar to the Bailey, but can be done remotely. So you don't actually have to have the kid in clinic with you. And see is it as good? And then if it could be an outcome measure, then we could potentially avoid a lot of the travel to sites, which would be huge. Same for parent-reported outcome measures. We have to see what the data looks like, but if they are very nice, reliable trajectories, could we rely a little more heavily on that and seizure diaries that could be gathered remotely as well?
And or the net, which we're validating with Tom Fraser, and or the ORCA, which is observer reported, and or some kind of headband low electrode count EEG monitoring, right?
Yeah, all of these things I think
And
is,
the actimetry too would be potentially something that could be sent to a
Yeah,
Yeah.
good
person
catch.
remotely.
That's a great point.
yeah.
So exciting. We're so lucky to have people like Dr. McKee and Alicia Harrison. There needs to be some kind of honorific before GC's names. You're not doctors, but you're just underappreciated badasses. I mean, we're so lucky to have you guys and the CHOP team working on this. Thank you so much. No more questions? We have gone over.
Yeah, thank you so much for your time. I know that your time here is could be spent other places, and so we really, really appreciate it. And thanks for everybody for watching today.
Of course, for having us.